Test, test, test. Cool. Alrighty. Got that going on Patreon. Got it going here. Just got to share my screen and then we can get started. Window. All right. Hey everybody, I'm Patrick the Biochemist. Today we're going to be bringing this paper right here, SARS-CoV-2 peptide fragments selectively dysregulate specific immune cell populations via Gaussian curvature targeting. So that's quite a mouthful of a title. For people that are not in the sciences, this is like, you're going to look at this and be like, oh my God, this is a headache. What is this? So starting from the top, how do we get to this? We just type this title into Google, and then it'll pop up as the first thing. You go to the Journal of Penis. Penis. You know, you got to have fun in science, or else, you know, everything just not worth doing. You gotta have a sense of humor. So the Journal of Penis, PNOS, which is Applied Physical Sciences. That's the acronym for that. So I don't know why they didn't do APS, probably already taken by another journal. But they decided to go with penis. And all right, enough jokes. But just looking at the title, it's sort of, we have to break it down part by part. So SARS-CoV-2 peptide fragments. So this is probably talking about the spike protein, how this is something we found out from very early 2020 during autopsy studies that everywhere throughout the body, they were finding spike protein viral fragments. So they were finding it in the brain, the liver, the kidneys, the eyes, nerve tissue, the muscle tissue, really just everywhere. And people that had COVID infections and died because they're doing the autopsies on bodies to figure that out, like what was going on with this virus, what it was doing, back when we knew absolutely nothing about it. And during these autopsies, they found spike fragments everywhere, which is the little protrusions that come off of the membrane of COVID. So that's what I'm guessing it is. So one of the things I pre-populated here. So first, what is a peptide bond? So in general, peptide bond is sort of a connection between two amino acid groupings. So it'd be a carbon to a nitrogen connection. That would be the peptide bond, or I guess the dipeptide bond here. And so how is this relevant to COVID? So if we look over here, there's a fusion peptide. I'll try and zoom in so you guys can see this. Let's see if we can blow up the image in a open image in new tab. Then we can zoom in. So right here, it's the S1, which is the spike protein subunit, the purple right here that I was just telling you about. And then connected to that, there is a fusion peptide. So when we say SARS-CoV-2 peptide fragments, so we're saying probably the breaking of this bond and then the spike protein fragment that's left behind after a COVID virion, a COVID virus dies, is killed for whatever reason, just like natural cell death, did its job and is replicated and produced offspring within your body. Really just that. So this is what I'm thinking it is. I haven't started reading the paper yet, obviously, because I do all this live. I don't pre-populate anything. I just went from the title. I was like, oh, might be beneficial to talk about peptide bonds and then also Gaussian curves, just because that's literally what the title was saying. So this is what I'm thinking that we're going to end up talking about in this paper, is the breaking of this peptide bond right here, and then the littering of the spike protein throughout the body, and then how that triggers the immune system. So that's what my guess is. And then they're talking about specific immune cell populations. So maybe it's going to be T cells, B cells, I don't know, all sorts of different types of immune cells. I should have worn my immune cells of the immune system t-shirt. I should have done that. But we have that. And then there is Gaussian curvature targeting. So if you haven't taken a math course before, or statistics course, you probably have never heard of Gaussian curvature. So there's really this could fall into two different types of cams. They're in calculus, basically the same thing. But first, there's Gaussian statistics, or so that one curve could be like the normal curve that we call a Gaussian distribution in statistics. So that's basically saying that if we have like a population of 100 people randomly, and we measure everyone's height, for instance, just some sort of variable, then you're going to have sort of most people falling within a certain proportion of this mean height. So for men, maybe like 5'9", and you'd have most people falling within like 2 inches of that. So anywhere from 5'7" to 5'11" for the most common height for men. And then the taller you are, or the shorter you are, the less in this random grouping of people you will have people that are the same height as you. So if you're very short, like 4'11", or very tall, like 7 foot, there's probably only going to be a handful of you, like just yourself, maybe one other person. And that's sort of what it means for like a Gaussian distribution curve, is just the it's basically saying if you take a random distribution of any population that's representative of that group for whatever you're measuring, you're going to have this distribution where you have a middle value, a mean, and then a certain proportion of values from that. You start to see a die off where there's not many of whatever you're measuring at the high end and then at the low end. So that's Gaussian statistics. You could also have Gaussian curvature. So this is another thing. This is what I think they're probably going to be talking about, because like both are Gaussian curves, but I think this is what we're going to be talking about, surfaces. So in calculus, we talk about basically taking a two-dimensional plane. So if you think of like a parabola, and you take that parabola and you spin it in a circle, then you would get a bowl, sort of like this right here. So if you took a sideways parabola and then you spun it around, it would create this shape right here. Or if you took like a straight line, you spun it around in a circle, you would get a tube. And if you took like an inverse parabola, spun it around in a circle, you get a sphere. That's essentially all this is doing. So how you could do this, you could say we're looking at a very specific area. And within this area, this surface area, because this is surfaces, that's all you're really doing. You're looking at a two-dimensional plane spun into a three-dimensional axis. And you're basically using just like simple formula, simple curves to sort of define the surface within the shape. So if you think about tubes like this and this, then you could think about maybe like blood vessels is what this would be representative of. And then you could talk about the amount of maybe like immune cell interaction, or like the number of spike protein fragments within this space. So like the higher number of spike proteins, maybe the higher immune reaction, the lower number of spike proteins in that space, the less. Maybe that's what we're going to talk about in this. But just to give you sort of like a high level basis of what we're probably going to be discussing within this paper. So again, probably talking about spike protein being broken off from COVID after, for whatever reason, when the virus is killed off, leaves behind a bunch of lingering fragments that our immune system isn't very good about cleaning up, apparently. We've known this since very early 2020 in the first few cases of COVID where people died during autopsy studies. So probably breaking this peptide bond right here. And then we're probably looking at the area of small surfaces in a subsection, probably looking at like blood vessels, just the amount of immune cells within that blood vessel within a certain area is probably what we're looking at. But again, let's dive in. So again, you want to, whenever you pull a paper, you're going to download the PDF, and then always look for any supporting information. Sometimes it'll be called supplemental information. Sometimes it'll be like supplemental figures, supplemental software, supplemental just data, like data that didn't fit the narrative, but is still important for people to have if they want to do their own analysis based off of the work of these researchers. So you can just like click on that. It'll bring you down to that area, and you can download the appendix. So I already did that. So this is the paper right here. And then this is the supporting information for that paper. So yeah, let's dive in. Do a quick check. Nope, no comments yet. Okay, so these people are out of University of California, Davis. Is where the paper is from. Oh wait, that's the editor. Weird. Usually they have the is it down below? Ah, here we go. So the authors. Huh. Here we go. University of California, University of California. So there might be a minor conflict of interest here because all of these people are University of California, are the authors, but then the editor of this journal is from California, University of California. So that's weird. So they probably know the people directly that they work with that submitted this paper. But let's judge them by the quality of the work, not because they just happen to work at the same university. You know, small fields, everyone knows each other. This one, the editor of the journal just happens to be at the same university as the people in the paper. So it doesn't really matter. All right, diving in. So this is the abstract right here. So immune cell populations are dysregulated in COVID-19 for currently unknown reasons. Plasmacytoid dendritic cells populations are reduced, thus hampering antiviral responses. So this is a very specific type of immune cell. The dendritic cells are typically ones that can differentiate into other types of immune cells once they get exposed to an antigen like the spike protein. So they're saying that this sort of like core immune system cell is getting reduced for some reason during an infection with COVID, and this is preventing because like that's like the initial starter cell. It's like here, let's produce like killer T cells and B cells so that we can attack this thing, whatever virus or pathogen is infecting your body. So if you like have for whatever reason you're not producing enough of these, or these are getting infected themselves or getting killed inadvertently through the COVID infection, this is going to reduce your ability to actually fight off the virus in the long term. So not only that, but CD8 T cells, these are the killer T cells, populations are reduced, the levels of which has emerged as an index of disease severity. So this is all during the acute infection when you're actively infected with COVID. And recent work has shown that the proteome of SARS-CoV-2 is a rich reservoir of antimicrobial peptide-like sequence motifs, xenoamps, which can chaperone and organize dsRNA for amplified toll-like receptor 3, mediated inflammation in vitro and in vivo. Here we demonstrate the proteolytic digestion of the SARS-CoV-2 spike protein by host trypsin-like serine protease directly produces xenoAMPs, synchrotron, small angle X-ray scattering, mass spectrometry, and theoretical analysis based on continuum membrane elasticity show that the proteolytic generated xenoamps from SARS-CoV-2 proteins in vitro and machine learning predicted high-scoring xenoamps all induce negative. Did I skip a line? SARS-CoV-2 proteins in vitro, machine learning predicted high-scoring xenoamps all induce negative Gaussian curvature necessary for pore formation in membranes. So what they're doing here, so this is very, very dense. And this is also working at the fringe of my knowledge base. So they're using X-ray scattering. So this is one technique where you're going to shoot X-rays at something, and then those X-rays that bounce back will give you an image of these small surfaces. Mass spectrometry. It's when you sort of dissolve a small amount of something and then look at all of the components that that thing is made up of. That was a very loud car rev outside. Um, and that gives you sort of all the components that something's made up of, so you know what that is. Oh my goodness, my screen did not change. 1 second, let me share that. There we go. Now you can see what I'm talking about. So small angle X-ray scattering, that's going to show you sort of like the surface characteristics of something as an image. Mass spectrometry, you basically like dissolve something down into its core components, and you see how much that is made, that each like component is made of, and that gives you a sense of what the thing actually is. And then they're also using computational modeling, so they're doing some math to come up with to generate like what the how springy the membrane is, how it's able to survive or not. Yes, that's much better. Sorry about that. That was my bad. I was just like, I just like glanced back over at my view of like what you guys are seeing, and it's just like, boom, you guys can't see anything. So they're doing that. And I guess they're looking at like how the membrane responds to different things. Is what I'm getting so far. So we find that the xenoamps alone, as well as the xenoamps synergistically with endogenous AMP-LL37, can induce negative Gaussian curvature in membranes, a computational analysis of immune cells with morphologically complex shapes. So they're looking at those dendritic cells, the CD8 plus cells, the CD4 plus T cells, suggests that surfaces with high local negative Gaussian curvature can concentrate AMP-like sequences and promote selective membrane disruption. So I'm not sure from this if they're, so what they're saying is like, so you have like a positive membrane would be sort of like a nice spherical shape, and then a negative would be sort of a contracture over here. So we're starting off with like a positive shape, and then we're getting sort of like a contracture like this, where it's still connected. And that contracture can then result in rupture of that membrane. So then the cell would die. Um, and consistent with this hypothesis, experiments with freshly isolated human peripheral blood mononuclear cells confirm that viable plasmacytoid dendritic cells and dendritic cells and T cells are significantly depleted after xeno-AMP exposure, in contrast to monocytes and neutrophils, the immune cell subsets with spheroidal morphology. So that's just saying spheroidal is again like that circle membrane that I'm talking about. And then they're saying that because of these so SARS-CoV-2 is interacting, creating these xenoamps. Don't really know what those are. We can look it up in a second. It's creating these protein structures from COVID proteins. When that interacts with the any of these three types of immune cells, the plasmacytoid dendritic cell, the dendritic cells, and the T cells, it's causing them to change shape. And this change in shape is it's possible that those cells will then sort of break and die. So it's directly something from the COVID that's being produced is directly causing damage to these three types of cells, which is causing a lower level of those in your body. But there are two types of cells that seem to not be impacted by this, which are monocytes and neutrophils. So these are sort of your passive immune cells. Neutrophils help with like clotting and stuff like that. Monocytes will go around, they just kind of like gobble things up that shouldn't be there. So those seem to be unimpacted. But the other types, the ones that like actively, selectively go through and specifically target cells, the T cells, the dendritic cells, which are the ones that sort of present the antigen to like the T cells and the B cells to be able to go kill things, and then the plasmacytoid dendritic cells, which, let's see which ones those are, because I'm not familiar with that one. Plasmacytoid dendritic cells. So they amp up type 1 interferon. So they specifically look at detecting for foreign DNA and RNA. So they would be like detecting the like mRNA from the COVID, or the RNA from the COVID. The mRNA is what we use for vaccines. RNA is in the COVID. And then structural data from the Omicron variant, xenoamp homologs. So they created something that, so instead of like having COVID create these things that are then being like introduced to the immune system and then causing this changing of shape and then death of these cells, they're creating their own similar feature. So taking the COVID out of the picture, they create a similar feature, this xenoamp protein, which I'll Google in a second so we can see what that is. And they're having that interact with the immune system. And it's also resulting in these changes as well. So it shows when you take away like the COVID and what it's producing to cause this, and you create something new that is the same thing as what the protein that COVID is producing that's causing this issue, and that thing that is new that you're creating that to be similar to this but is not this, that can also cause the issue, then you're showing that this is in fact what is causing the issue. Is sort of like how scientists think about that. So let me look up xenoamp or xenoamp. Because again, this is on the fringes of my knowledge base. It's way out there. Two distinct concepts. Oh, I'm way zoomed in. So, yep, let me switch again. For some reason, my window. Oh, here we go. I got to share the entire screen. That's why we weren't being able to switch back and forth. So xenoamps refer to two distinct concepts, set of SARS-CoV-2 viral peptide fragments that trigger inflammation mimicking host antimicrobial properties, or an older minimalist. Okay. Xenoamp peptide fragments. Oh, redirected me back to the paper. So we're actually going to see an image further in the paper. So let's see if we can shoot to that image, because I think that'll help explain. Well, I didn't. Oh, there it is. Figure 3, comparison of membrane remodeling capacity between COVID, amps and histone. So I guess what it's doing, it's basically a protein fragment that is, so I'm guessing xenoamp S. So it's maybe not xenoamps, but xenoamp S, which is probably, like we were talking about before, the remnant of the spike protein with the peptide bond that's still sort of active, electrostatically active. Um, oh, got a. Do you need me to re-explain something? Sorry. But essentially, what's happening is it creates this. What I'm guessing is remnant of the spike protein that is still when you break a bond, it still has a charge to it. So when that charge then interacts with the membrane, so we're thinking the membrane has slightly positive charge. This might have a slightly negative charge that creates an attraction. So then it'll bond to the surface of the membrane, and as you'll see here, it's pulling the top of the cell, bottom of the cell together, which is causing that contracture, causing it to change shape from like a sphere to sort of like an elongated um, counter. Um, let's see if I have something to show. So instead of making something like this, it's going to create something more like this. And when you have this sort of like pinched outer curve, here we go. When you have this like pinched outer curve shape like this, that's going to have very high stresses down the middle and in the back, which is going to make it so that it's more likely for this thing to then tear. And then when you have a torn membrane, things just spill out and you die. So that seems to be what's happening. Okay, so then we can talk about the significance and then go into the intro. So they say the significance of this is that previous work has demonstrated that the proteome of SARS-CoV-2 can potentially be a rich source of AMP-like viral fragments, which are associated with severe COVID-like inflammation in vitro and in vivo. So what this is saying is that the proteins from COVID can produce these fragments, these proteins, which then get littered throughout your body. They've shown this occurs both in cell cultures, like away from the body, which is in vitro, or in vivo, so in people as well. And then here they demonstrate directly that proteolytic processing of SARS-CoV-2 proteins can yield xenoamps. So they showed here, sort of like we were showing in that image before, that breaking this bond can create this new protein, which can then injure the immune cells of the immune system. And that full heterogeneous ensemble of resultant fragments can collectively exert AMP-like pore formation activity. We described an unanticipated general mechanism of host cellular targeting for viral AMP pore forming peptides based on local Gaussian curvature of the host cell membrane. So yeah, so this is I thought it was going to be like cell walls, and they're going to be looking, or not cell walls, like they're looking at collectively fragments within veins. So joking aside, not necessarily die, but inflammation from these assaulted cells. So it would be local targeted cell death of these specific cells. So the dendritic cells, the plasmacytoid cells, and the T cells. So when this protein from COVID bonds to the membrane, it will actually kill those cells, which causes, if you think about it as like a whole population of these cells within your body, you're going to have less of those. So it's going to be actually directly targeting, damaging, and then killing those cells. And that's what's causing this reduction in population. And when you don't have enough of these cells, then you can't produce an efficient cellular like immune response. So you're going to be infected. So with COVID, that means your infection is going to last longer. And again, like once the COVID infection is fought off from your immune system, hopefully, then this reduction is still going to be present. Your body's going to have to recover and build these things back up, which takes time. But it means that you're going to be more susceptible for a while, like we've already seen, to like other infections as well, not just COVID. So we describe an unanticipated general mechanism of host cellular targeting for viral and pele-like pore peptides based on locally and Gaussian curvature of the host cell membrane, and show that this mechanism can selectively target and deplete specific immune cells in a manner consistent with clinical observations for severe COVID-19 patients. And the reason they're focusing on, so everything I said, that's like the fancy way of saying it. The simple way of saying it is what I just said to you, is that COVID, when it breaks down, when the spike protein pops off, it creates these proteins that can then bond to your immune cells, causing them to change shape. And when cells don't like to change shape unless they're like specifically creating a bond with something to cause some other type of reaction, and then they'll go back to their normal shape after that, or they'll be permanently changed, and then their job will be done. But what this will do is, so these proteins just sitting there, interact with your cell's immune system, these specific three, and then it causes them to change shape, rupture, die, and that reduces your overall population of that, makes it harder to fight off infections. It's going to be the most pronounced in severe COVID patients. This is why they're focusing on severe COVID patients, but it's also going to be something that occurs with every single infection, whether it's asymptomatic, mild, moderate, or severe. But if you think about it, like with each infection, we know that there's a higher viral load, the more severe your acute phase is. So that means the worse your symptoms are, the worse the infection is, then the more of these fragments are going to be produced, which means there's going to be more of these interacting with your immune system, which is going to result in a lower cell population of these immune cells, which is going to make it harder for you to fight off that current infection, as well as to then fight off future infections. It's going to make you, for lack of better words, immune compromised from that infection, particularly like in the higher, like the moderate, the severe type of acute illnesses, is what that's telling us. So in most viral ones, this is going to be super dense. Yeah, mask. That's what that's. But this is like direct cellular evidence that they're showing to show, like people like AJ Leonardi predicted this, like mental model theories, several years ago. But because everyone's continually getting infected and continually doing that at a population level, if we take this small nugget of information that we just found out just from this, barely even scratching the surface of this paper, it means that like population wise, our immune systems are becoming slowly weaker and weaker and weaker over time. So we're becoming more and more susceptible to infections, not just COVID, but everything else, because we don't have this ability, like where our reservoirs, our stockpiles of these immune cells are slowly depleting with each infection. Where again, like if you have a worse infection of COVID, you're going to have more impacts. But if you have like a milder one, you're not going to have as much, but you're still going to have these impacts and reductions. So if you're getting infected 2-3 times a year, this is going to stack up cumulatively over time. So in most viral infections, appropriate molecular and cellular responses are orchestrated to achieve viral clearance. This is just saying that we need all these cells interacting together to sufficiently clear viruses. Plasmacytoid dendritic cells detect viral components and initiate innate responses, including type 1 and type 3 interferons and cytokines to activate adjunctive immune cell types such as CD4 plus and CD8 plus. So that's sort of what I was saying before. So these plasmacytoid dendritic cells are sort of like the initial starters that kickstart like your CD4 and your CD8, which are your killer T cells, the ones that'll go out and initiate these focused targets on whatever you're being infected with, because it'll present like this is the antigen that we want to take, go out and fight and destroy everything that looks like this. And your B cells too, the memory B cells. So antigen-presenting cells display viral peptides, activate T cells, and prime them for killing of infected cells. This is called CD8 plus T cell cytolysis, which is basically what I just told you. In turn, monocyte macrophage populations expand. So these are the ones that are sort of like the degenitors of the body, the macrophages and the monocytes. They go around and just gobble up cellular debris. So the CD8 are kind of like the knights. They'll go around, they'll go smash, 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 everything that looks like this that the dendritic cell showed me. And then they leave behind a bunch of fragment remnants, and the macrophages become greater population. They just go gobble, 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 take all those things and dispose of it. That's sort of like the sequence of this. So when when the virus is actually killing off these cells that are presenting the antigen, causing these other cascading things, then you're going to have less of an immune response to that thing, a less targeted immune response, because you'll still have like your passive immune system that's fighting it off, that you'll just like have like sort of sentries walking around and then like bumping into things being like, I don't recognize you, kill, kill, kill. But our targeted one that goes around the knights that fight off everything, they're not going to be as active if they're not given instructions to do that. So in turn, monocyte macrophage populations become strongly activated to promote antiviral defense. In several cases of COVID-19, this orchestration of protective immune responses is drastically different. Innate responses are dysregulated, and neutrophil-mediated inflammation is perfusive and protracted. That's interesting. So I guess it's also saying that the neutrophils, so the inflammation you brought up before, so not only we're having this like actual population just being destroyed, we're also, because like inflammation is a typical part of being sick. That's why we'll feel like swelling, stuffy, all over. So it's saying it's perfusive, so it's widespread, but also protractive, that it's not as significant or sufficient as it typically would be, which is sort of a weird competing thing. So I guess we'll have to read more into that because I don't get how it's both of those things. Resulting in an off-target inflammation that damages by, okay. So I guess what it's saying is that the neutrophils, because they're like everything's not given as much guidance as it should be in your immune system, it's sort of like your body's like, something's wrong, we're fighting it off. And then instead of like being like targeted to fight off the specific virus, like the COVID infection that you're being infected with, it's going, I don't know what to do. I'm just going to attack whatever I think I need to. And it starts like attacking all sorts of things that it like your own tissue, your own cells, because it's like, I got to find something to kill. So I guess that's what it means. So it's protracted in the sense that it is not targeted to like the COVID virus, but it is perfusive in the fact that it's like freaking out, going, I need to destroy something, something's wrong. And it's sort of hurting your own body. It's like being like, that cell is kind of off looking, I'll attack that. And it's wrong in its assumption. Like the cytokine thing. So very early on, they're like proposing like, oh, it's like a cytokine storm. Like all of a sudden our immune system is kind of going off the kilter. It's something's happening that's causing it to just like attack everything. Well, this is sort of like that. This is a more refined version of just like blaming the cytokine storm, because like that's something that people do in academia a lot, is they'll just be like, oh, cytokine storm, things have just got dysregulated and everything goes haywire and just kills everything. And it's sort of like a cop out. It's what people in science will say when they don't understand something, is they'll blame it on the cytokine storm. They'll like think of like osteoarthritis. We don't really fully understand exactly how everything works. We know that there is inflammation and this progressive thing to generate tissue and cartilage within that local area, but we don't really know exactly how it happens. So we call it, oh, it's just the cytokine storm. It's kind of like a hand wavy thing that's like something that's well known in science that we don't fully understand. We understand it to a certain extent, but not exactly how it works. So when we don't fully understand something, a lot of times the scientists will just be like, oh, it's a cytokine storm, and that's what's causing it. It's just like a brush it under the rug type thing. But this is saying, oh, this is what's happening. And then because like your innate immune system, the one that just kind of like the sentries that go around and trying to kill everything, inadvertently, like they know something's wrong, but they don't know what is wrong. These ones are sort of going out of control because they're not getting like the full direction that they're supposed to. So a profound alteration is also observed in specific host immune cell populations, lymphocyte including CD4 plus T cells, CD8 plus T cells, B cells, and NK cell counts are significantly reduced in peripheral blood in COVID-19 pneumonia for both mild and critical cases. So again, this is what I was saying before, like they're focusing on severe cases because that's the most impactful that you're going to have, because like the more virus, the more of these little peptide bonds that are going to go and attack, not peptide bonds, the AMP-like bonds, the proteins that are going to go and attach to things and end up causing these issues. But it's also going to happen for mild cases too. But again, you're going to have more instances of it because more virus and the more severe cases than the mild cases. And they're saying that this is so commonplace that CD8 T cell count has emerged as the most robust indicator of COVID-19 disease severity. So an interesting implication of this. So we know that the COVID vaccines, like there's a lot of things that go into it. So like the reason like the vaccine effectiveness has been reducing over time, a lot of it has to do with timing, frequency. Barely anyone's getting vaccinated, but for the people that are, we're still seeing vaccine effectiveness wane a lot faster than it did before. So an implication of that might be that because like the last time they did a actual estimate on this, like 2.5, 3 years ago. No, I think it's like 2.5 years ago. They estimated about 90% of the population had had at least one COVID infection. Mike Horger has an average on his PMC chart that it's like 3.8, I think, last I saw, infections on average in the past 6-7 years. Because we completed year 6, we're going into year 7 of our ongoing pandemic. So it could be part of the reason why the vaccine effectiveness is waning so fast is because we're having basically a depletion of our immune cells, so we're not able to mount as efficient as of a immune response to like vaccines than we should. It could be tied into that, but there's also like we're picking variants that are too far back, not like more recent and circulating in the current time. We're not getting vaccinated frequently enough. We're not vaccinating like the entire population every single time. The most recent vaccine, the 2025, 2026, only like 17% of the population has had that. That for adults, for kids, it's like 8.6% of the population when I just looked at it the other day. So like we're not getting enough vaccine. There are a lot of other factors, but this could also be a factor that's playing a role, I guess is my point, which is interesting to think about. And similarly, rapid depletion is observed in plasmacytoid dendritic cells subsets, which normally act as first responders in viral infection through fast and potent early interferon signaling to activate downstream antiviral interferons stimulated genes, ISGs. Such cellular depletion can persist for up to 7 months in both hospitalized and non-hospitalized patients. So that's the kicker right there. If we're getting infected 2 to 3 times a year on average, and these effects in our immune system last for about 7 months, because when we say in a study up to 7 months, that means the study was typically performed out to 7 months. So it could persist longer than that time period, but we can at least say up to 7 months. It's a weird thing in science we do. We say, because studies have to take a certain amount of time, we can only say like, oh, our study went out to 2 years. So we can say up to 2 years, we have this issue, but it could persist longer than the 2 years. But in science communication, we're not very good at saying like, yeah, it could go longer. We don't know. This is what the study window was. So we can say up to 2. So in this one, up to 7, it could be longer than 7, but we can say at least up to 7 months. But if you're getting, again, infected 2-3 times a year, this is going to be a compounding effect that happens year after year after year. And we're not recovering enough between infections to then restore our immune responsiveness, if we can even restore our immune responsiveness. I'm assuming we can, particularly in like younger, healthier people, as opposed to like older people. So like in a population curve, younger people are better at mounting immune responses, older people not so much. So like these effects would probably mount the most in people with active immune systems than less active immune systems. So we're probably going to see, like we have been seeing, younger adults like between like 18 and 35 are probably going to have the most impacts quickest, but everyone is going to be impacted and having these lowered responsiveness particularly for people that are getting infected multiple times a year. Yep, and then overlap, exactly. So in contrast, monocytes, so when you have really dense papers, you have to like think about it a lot and think about it like that. So it takes a really long time to go through papers like this. I'm already at 44 minutes and I have not even gotten past page 1. So in contrast, monocyte populations were not significantly different between COVID-19 patients and normal healthy individuals, despite extensive research. So again, like they're saying, like it's not impacting all immune cells, it's impacting like some immune cells specifically, but like this is all like an orchestra, like many things going on at the same time. So when you're having a reduction in one thing, it's also going to impact these other things just indirectly, which is why we're seeing like those like the cytokine storms, like the indirect immune responsiveness to viruses and stuff. So despite extensive research on COVID-19, understanding of the mechanisms driving the selective deficiency of antiviral and immune cells remain incomplete. Recent work has shown that the proteome of SARS-CoV-2, but not those of common cold coronavirus, is a rich source of xenoamps. Yep. So this is just saying like SARS-CoV-2 is unique. It's a novel virus. We're not seeing this with common cold coronaviruses. So even viruses that are very similar, like sisters viruses, like coronavirus 1, or more like common cold coronaviruses, they don't really produce this xenoamp stuff. It's specific to SARS-CoV-2, which again, novel virus, so we have to treat it like it is, like because we don't know. We're still learning stuff about it as we are right now. So once released from the viral protein during proteolytic degradation, so these are produced once the cell dies. So this could be just like natural cell death, like once it replicates, the prior one that tried to cause this replication within other cells within our bodies, then that one dies, leaves behind fragments. So this is just a natural part of the COVID life-death cycle within our body. They can chaperone and organize DS. So DS is double-stranded RNA for amplified toll-like receptors activation, resulting in elevated pro-inflammatory cytokine secretion in diverse cell types and culture. Epithelial cells, endothelial cells, monocytes, and macrophages. A COVID-19-like transcriptome from endothelial cells is a significant boost in plasma levels of interleukin-6 and CXCL1 in mice, while protease activity can, in principle, degrade viral proteins and thereby, in principle, suppress infection process. Certain protease families are found to promote infection. So this is something looking at like, all right, so how can we stop this from happening? We can use sort of like protease inhibitors, so things that can prevent these proteases from forming. But they found that when they tried to target this, in some cases it kind of worked, but in other cases it actually made the effects worse. So it's not going to be a simple thing, is what they're saying there. Because it's like here they're saying like in certain protease families they were found to actually promote infection, promote the propagation of the COVID within your body, and then to infect others. For example, the KLK family of trypsin-like serine proteases were found to facilitate influenza and beta coronaviruses, viral entry by cleaving and activating the viral fusion protein. A general picture of how proteolytic products of viral proteins interact with the immune system, however, has not been systematically investigated. So this is saying they're like, hey, we know that this is a big gap in the literature. This is something that we don't really understand at this point, but it's something that's worth investigating, and that's why we're looking at it, is what they're saying. And then here we examine the possibility that a subpopulation of SARS-CoV-2 viral fragments generated from a host proteolytic processing can selectively suppress specific immune cell populations via an unanticipated form of host cell targeting based on detection of membrane Gaussian curvature. So again, they're looking at the curvature of the cells and how they change with the interaction with these xenoamp sequences. So these viral fragments from the COVID infected hosts are expected to be heterogeneous and correlated to specific enzyme proteome enzyme of the infected host. What's more, since many amps preferentially permeate bacterial membranes over mammalian membranes, higher local amp concentration are in general required to impact host cells. The studies here are designed to be a first exploration of these hypotheses. Using a combination of liquid chromatography with tandem mass spectrometry and machine learning, we found that viral fragments generated by a tryptic digestion of SARS-CoV-2 spike proteins include AMP-like sequences The propensity for xenoamp production from viral proteins is enhanced by the enrichment of cationic residues in the SARS-CoV-2 protein. Since cationic charge is one of the defining characteristics of EMP sequences, the process of xenoamp generation is cognate to the production of human EMPs, as exampled by the activation of human EMPs by the protease kallikrein, KLK5. I guess like the important part of that, like that was a bunch of mumble jumble, is this part right here. So our bodies are not just like our cells, but we also have bacteria, gut bacteria within us. So in certain areas, it seems like the gut bacteria that we have will actually COVID will be drawn, like these viral fragments will be drawn to that instead of our cells. It has a preference to those bacteria over us. But again, that can also cause like gut dysbiosis, like you'll disrupt like the flora within your gut microbiome because of this. And we've seen that like people get a lot of like diarrhea and upset stomach, nausea. We call that like GI. We think about that as like GI focused COVID issues, like long COVID or post-COVID complications, but that is something that happens. It seems it has a sort of a preference for these, but if again it sort of permeates everywhere within our body, so you're not going to have bacteria everywhere within your body, so it's also going to attack our cells as well. Using small angle X-ray scattering and molecular dynamics simulations that xenoamps can mimic the ability of to induce negative Gaussian curvature in lipid membranes, which is a geometric requirement for forming transmembrane pores. This membrane remodeling ability is observed for xenoamps generated in a broad range of distinct contexts, including specific xenoamps generated from direct tryptic digestion of the SARS-CoV-2 spike protein, synthesized xenoamps that exhibit high predicted it's not theta scores, omega scores, I guess, in our validated machine learning classifier for identifying AMP-like sequences in the peptide mixture released during trypsin or KLK digestion of spike protein. Xenoamps can induce non-Gaussian curvature and can enhance the pore-forming activity of endogenous AMP LL3 zone as a division of labor mechanism consistent with these results. Radial diffusion assays indicate that these COVID xenoamps have been broad spectrum antimicrobial activity against all these different bacteria and some fungus with antimicrobial levels quantitatively similar to typical AMPs. Using mean elastic theory, we estimated a xenoamps induced transmembrane pore size of 1.46 to 1.86 from sex measurements size, which is comparable to that generated by AMP-like N-terminal fragments of histone, shown to be capable of inducing lytic cell death of endothelial cells. So that was again a mouthful. This is again a very dense paper. It's saying that the holes that they've shown that it'll generate holes. These holes in other studies have been shown to cause lysis of the cells, specifically endothelial cells. So epithelial is our skin, endothelial is the lining of our blood tissues, our blood cell, like our veins and stuff like that. Which again is interesting because we've seen endothelial damage of our the blood vessel walls. So this is probably how that damage is occurring, is through this. So it's causing lysis, which is splitting, which is rupture of a cell. So these holes that are being generated from like the changing of shape is causing lysis, which is just explosion of the cell, rupture of the cell membrane, and cell death. To again answer that question from earlier. And they also showed this in computer modeling. So not only do we have like another study that showed that these pores that are being produced that they saw with their imaging techniques and also their modeling software, it all shows that this is what's happening. So this again is actually a very good study, very, very dense, which is why we're going so slow through it, and also fringes of my expertise. We propose a model preferentially partitioned to membrane regions with strong local non-Gaussian curvature, such as the septum of dividing bacterial cells, the microvilli-rich morphology of activated T cells, or the spiky morphology of activated polycytolic dendritic cells. I don't remember the word, but I don't feel like looking it back up. Last, we directly examined the effect of these SARS-CoV-2 viral xeno amps on different immune cells that are freshly isolated from human peripheral blood. So they're taking these viral fragments. Lysis, yeah, lysis is LYSIS. It's when you have splitting of a cell membrane. It's a specific word for that. Is cell lysis is splitting, damage, rupture. Here I can show you an image of that. It's a very common thing, cell lysis. So like we have like a rigid wall typically for whatever cell we have, and then rupture is lysis is when it splits like this. LYSIS, yep. And then so what they're doing is they're taking these viral fragments from the COVID. They're taking fresh immune cells from a person's blood, and they're taking these fragments, having them, mixing them in with the immune cells and seeing what happens is what they're essentially doing right here. And then again, they're reiterating what they found before is that they're affecting these polycytolic dendritic cells, the CD4, CD8 T cells with a very high efficiency. So like comparatively, they're these CD8 T cells are being damaged at a 3.2 to 64.5 times higher cell death. So like that's really high. This is not like percent. This is time. So like if you have like a baseline of like 1 times cell death, this is 3 to almost 65 times greater than that baseline of the other cells. So it's really specifically targeting these cells, which is not good because these are the ones that specifically will target viruses like COVID to kill it, which makes it harder for us to clear a virus if we're not having our specific killer cells for that virus. Oop, didn't mean to highlight. But not the more spheroidal cells that don't seem to be interacting with it, with this viral fragment. The monocytes and neutrophils don't seem to be impacted by that. Really good because if we have like low neutrophils, we're going to have a hard time clotting. So we would, if we had like any sort of like damage, we'd bleed out essentially, which is a very ugly way to go. But again, this is a little more insidious. What's happening, because when you have these stack up effects over a long period of time, it's going to have very large population-wide impacts. We're going to, like we already are starting to see right now, we're going to see a resurgence of all sorts of different types of viruses and bacterial infections and fungal infections that we haven't necessarily seen for a long time. People are going to be scratching their heads going, why are we seeing all these things? We don't typically see these here in the US, and it's going to be because of this issue right here. So in a more general compass, these results suggest a general mechanism through which the host cells types with specific morphologies can have enhanced vulnerability to these viral fragments of the COVID. So tryptic digestion of SARS-CoV-2 spike protein produces heterogeneous fragments. So interestingly, I thought that this is just when the COVID like dies naturally, like it's releasing these viral fragments, but also when it's being digested by our immune system, it seems it also releases these fragments. That's sort of like the tryptic digestion right here. It's like when our cells start breaking down the COVID, it produces these fragments, which can then go on to cause these issues. They're just saying SARS-CoV-2 is an enveloped single-stranded positive sense RNA virus. The average number of COVID virions in infected hosts is estimated to be 10 to the 9 to 10 to the 11th virions, virus particles, due in part to their high copy numbers. COVID proteins that are observed at the highest concentrations include the spike protein, envelope protein, membrane protein, and the nucleocapsid proteins. So nucleocapsid is what we target for wastewater, and also for well, largely that's also used for our rapid tests as well. And then also we use it in, so the reason it's targeted a lot for that is because it's very stable. It's not mutating as frequently as the spike protein, but it is mutating. So it's making a lot of our testing less accurate over time because it is changing. Mutating means changing. The later of which binds to the viral genomic RNA that eventually assembles into mature virions. So the nucleocapsid proteins are what's inside the COVID. They're released sort of like when the COVID is dead, but they also help with creating replication of new viruses within your body. And viral proteins can persist in the host for months or even years in COVID infections. We've talked about that before in the past, as well as for infections for other viruses. These proteins are all in all, these proteins are all in principle eventually proteolytically processed by the immune system, including proteolysis and a severe service of epitope generation for antigen presentation. So they're saying we think, to our best knowledge right now, that when you have a COVID infection, you fully clear that infection over time, but it may take a long time for some people. And then they explore these interactions of these viral fragments, considering how the COVID protein is processed in order to assess the full spectrum of possible fragments from diverse serine proteases. We explore both human and non-human commercially available serine proteases. These include lyc, elastase, and cathepsin G, which have different substrate specificities. Lyc is a model trypsin-like serine protease derived from Lysobacter enzymogenes, probably. Let's see, Latin words. I'm not obviously dead language, but I've never studied it, so I always struggle a little bit with Latin. But Lyc's strong and predictable lysine-specific proteolytic activity facilitates identification of the broad range of potential peptide fragments produced from the considerable repertoire of trypsin-like proteases. So they're just using all these different things to try and dissolve the COVID spike protein, which produces these fragments and of different sizes and lengths. So it's this one in particular seems to be a good choice for that, for their studies. In contrast, elastase and cathepsin G are endogenous proteases stored in and released from host neutrophils, monocytes, and mast cells. Elastase cleavage targets C-terminal aspects of the small hydrophobic amino acids such as alanine, glycine, and valine. Cathepsin G cleavage targets C-terminal aspects of aromatic amino acids such as phenylalanine, tyrosine, and leucine. We examine how the SARS-CoV-2 spike protein, both the full-length version and S1 subunit, are degraded into peptide fragments when they're incubated with the above proteases. So essentially, they're taking spike proteins, they're dissolving them down with these different things to create these protein fragments, which then would interact with the immune system and potentially cause these issues. And this is their first figure. So this is sort of showing where the cleavage would happen. So you'd have it breaking right here, which would leave this fragment and this fragment, where this is the one that's probably going to be causing the issues. So this is going to be the yeah, this is going to be that spike side of the protein from the full protein. So that's what's going to be interacting. And then these are all the different lengths of the breakage, because the breakage is not going to be like completely even in every single thing, but it's sort of creating, again, that like Gaussian curve, that normal curve. where most commonly it's going to be sitting around, looks like between 12 and 20. AA is that angstrom units, like 10 to the negative 9. So very, very small. I guess I'll just sort of skim because we're getting kind of late into this. So let's see. Do my little speed reading. E, looking for E, schematic representation. But yeah, so this is how they're modeling it. You have like the cell membrane here, you have the outer cell, this surface here, the opposing outer cell there, and then the connecting components on the outside. It's creating pores, so these holes through that negative curvature, which sort of looks like this when it's going from this to this. So it's going from like a nice loose structure to a tightened structure that's swelled, which is resulting in large pores, which can then cause the cell to break. And then down here, they're saying finally we contrast the non-Gaussian curvature generating ability of the intact spike protein itself with that of the peptide fragments generated from the proteolytic digestion of the spike protein. Viral fragments released during proteolytic degradation comprise a diverse range of sequences, some of which exhibit physiochemical properties closely resembling AMPs, while others do not. Here we investigate what happens when cell membranes are exposed to the entire diverse population of viral fragments generated from proteolysis, which is composed of xenoamps and non-xenoamps, to see whether membrane remodeling activity is preserved. To answer this question, the spike protein is first digested into a mixture of peptide fragments. Oh, interesting. So it looks like when it has like a bunch of different sizes of these fragments, they can the smaller ones can actually go inside the cell membrane, potentially damage inside the cell. So not just like binding to the outside, causing that change in cell, those holes to occur. The smaller fragments, when they like took a bunch of different sizes of these like full spike proteins and then a bunch of different sizes of them through just like chopping at random positions, they found like the smaller ones could actually penetrate the membrane, go inside, and then cause damage inside the cell. So not only are we having this curvature change, which is causing these holes, which can cause cell lysis and breakage of the cell, but also the very small versions of these fragments can go inside the cell and disrupt things in there and cause internal damage. And that's, I think, what they're showing here from before. So you see there's like longer fragments, there's smaller fragments, and you see some smaller fragments going inside and causing changes inside. So I initially thought this was just showing like, oh, it's changing the curvature of things, but also it's showing penetration and internal damage of the cell as well. So this is the interesting part right here. So the monocytes, the ones where it's not bonding to them and causing these issues, is very smooth on the surface. So there's really nothing to like, I guess, attach to maybe. While on the T cells, there's a bunch of these protrusions. Um, so it looks like these protrusions can allow for the bonding of that, and then it'll cause those changes in the surface curvature, um, creating more of like a saddle shape like this right here. Which can then result in holes forming and then those cell breaks, cell wall breaks. And they're sort of using like a dividing bacteria is sort of an example of this. See how it's sort of going from like typically you would have like a dividing bacteria starts as like a circle and then becomes elongated, creates a saddle shape in the middle, and then that will then get smaller and smaller and then break and split. But that's a natural process for bacteria for dividing. But it's also a good representation. Like if you have a whole cell and the surface change becomes a saddle shape, so that would be the negative curvature, and then breaks, then that would just kill the cell, which is what they're saying and showing. So that's the interesting stuff right here. Oh, that's interesting. So they're looking here at spike protein xenoamps from the original like Wuhan strain, I'm assuming, versus Omicron, which is sort of what everything current is based off of. And from original Omicron, which I think is XBB, maybe I'd have to look that back up. So like several years back, we got the Omicron designation, and then scientists stopped updating it. Different story for another time. But you can see here that from the original Wuhan, there is much more cell disruption than there is with Omicron. So that's good. It's not doing as much damage. But again, the caveat to this is we know that there's more virus being produced. We're having higher viral loads with Omicron, even though we're not having as high of activation of these viral fragments. So because there's more of it, it can still cause this damage, which they've shown. Let me back up a little bit just to make sure I'm understanding. Non-xenoamps have diminished capacity for inducing non-Gaussian curvature in lipid membranes. Yeah, so Omicron was designated in November 2021. So that's how you know they haven't been updating names for a long time. Has high transmissibility, high number of mutations in the receptor binding domain. However, for reasons not entirely understood, the disease outcomes tend to be less severe compared to earlier strains of SARS-CoV-2. The Omicron variant spike protein, way back when, harbors 37 mutations. So it'd be interesting to compare that to like sort of today's variants on this exact thing. That would be something I'd be very interested in seeing. But again, this is like a early study on this idea. So that's probably what they're going to do next in their next paper. So technically, it looks like compared to the interaction with the cell membrane for these protein fragments, the original one was doing like 1.46 to 1.86 nanometer holes, while this one is 1.32 to 1.74 nanometer holes with a 10% variation compared to its predecessor. But again, what we saw earlier for the bacteria that they were looking at for the cell breakage, the cell lysis, this is still within that range. So while it's less curvature changes, it's still going to be capable of doing the damage that they're talking about. But what they're showing is that these mutations in Omicron, which truly only fully relative back to November 2021, potentially reduces the killing activity of the cells of the immune system compared to the past ones. So testing both Omicron and the original Wuhan protein fragments of the spike protein, they did the same study that they did earlier. So they took like fresh blood with immune cells from a person, introduced the Omicron ones, introduced the Wuhan ones to two different versions of this, and then they compared the effects. And for Omicron, it seems to be size dependent on if it's causing cell death. So the fragment size, it seems like the bigger fragments, the 30 micrometers, or sorry, micromoles size, micromole, I guess. Um, generated 68.3 times better, like more cell death than the smaller fragments. Oh no, sorry, that was xenoamps. So that was the original Wuhan. So Omicron xenoamps induced much lower death. So at this same 30 micrometers or micromole size of fragment, it induced 8.1 times greater cell death than normal, while the original did 68.3 times. So there's still like cell death occurring, but just not as much with the Omicron variants. But it's still overly targeting our CD8 plus T cells, so our killer T cells, even with Omicron, where it's doing 3.2 fold, 6.4 fold, 5.4 fold. Greater than the other cells compared to control. So still, we're seeing the same types of effect, but not as pronounced. So that's good, but it's still showing that we're having these issues, even if they've mutated away from this. But again, this is only relevant for like November 2021. We've had continued mutations down the line from there. So we'd have to look at current variants to see if the same types of things are occurring to like original Wuhan variant or current or like November 2021 Omicron. So that would be like the next natural step for these researchers. And Omicron, while it's not exhibiting strong toxicity, towards almost all of the immune cells except for monocytes. So that's interesting. That's different than the Wuhan one. So the Wuhan one didn't really affect monocytes, but it seems like the Omicron variants do affect the monocytes, the ones that are just going to go and gobble up the debris. And then they're saying the results presented here offer perspective on why the Omicron variant behaves differently compared to the native SARS-CoV-2 strain in certain human hosts. One distinct from viral replication and entry, because we know that SARS-CoV-2, the current variants have greater viral replication, better entry into the cells compared to the Wuhan variant. They have a higher efficacy in that. They're better able to replicate within us. But interestingly, these mutations also reduce the number of viral fragments that can cause these issues that we saw with earlier variants. But again, it's still causing these issues just to a lesser degree than it was in the future or in the past. And they're sort of proposing this as like a part of the feedback loop of between inflammation in COVID-19 patients and the production of these xenoamps, these spike protein fragments. They're saying that their previous work indicates that these protein fragments derived from SARS-CoV-2 spike proteins can organize double-stranded RNA into a liquid crystalline ordered complex, which amplify immune activation in multiple cell types, including monocytes, macrophages, epithelial cells, and endothelial cells, which exhibit a COVID-19-like transcriptome. Such inflammation can lead to the activation of trypsin-like serine protease activity, which increases the probability of creating more xenoamps. So they're saying that this can sort of create like the vicious cycle that we're seeing, that it might not necessarily, like it can potentially contribute to this like cytokine storm that people have talked about a lot, but through like a unique mechanism. So they're saying that like it can these fragments can bind to things, create more of like a changed structure within these things. Um, so liquid crystalline means making things more ordered, rigid, hard, um, which can also like when those non-specific immune cells are going around, they're like, that's different. That's not how it should be. So like if it's attacking like the blood vessel wall, your immune systems technically would be attacking the blood vessel wall. If this interacted with the blood vessel wall, caused this crystalline change, and that non-specific immune system saw that, is like, that's not normal, and then it attacks it and just destroys it, creating more viral fragments in the process, which then creates a cycle of these things binding to other things, changing the shape of it, our non-specific immune system going, that's weird, kill it, and then releasing more viral fragments of smaller sizes, and the thing goes on and on. And then recent studies suggest remnant viral matter from SARS-CoV-2 and from other viruses persist in hosts for months after their infections have cleared. SARS-CoV-2 spike proteins have been found to circulate in patients' blood for months in patients with myocarditis. These have been found in people up to years later. From early studies, like when in autopsies and stuff, they were finding it like a year or two after infection when people still had like a low number of infections. Persistence of viral matter in the host has been hypothesized to play a critical role or play a role in the chronic inflammation in long COVID. From the standpoint of the work here, the viral proteins can also serve as a persistent reservoir of these smaller like fragments of the spike protein being generated, which then can like bind to things and cause downstream issues, and may provide a possible explanation as to why the depletion of polys dendritic cells can persist for months after the initial COVID infection. Together with our earlier work, the results here suggest that viral fragments may contribute significantly to the phenomenology of COVID-19. In a more general compass, we hypothesize that there exists a virtual unexplored layer of host pathogen interactions mediated by proteolytic processing of proteins from viruses and microbes. So essentially, their ultimate conclusion in all of this is, hey, we found this really cool thing that these spike proteins, which we know are just circulating within people for a long period of time afterwards and just existing when our immune systems break those down. So first, like these things can bind with things, can change the shape of them, and that can cause our innate immune system to target that and destroy it, creating smaller spike protein viral fragments in the process, which can then go on to bind with other things, cause changes, and destroy cells, and specifically can bind with certain immune cells that are needed for specific immune responses, change those cells, kills those cells, reduce the overall population. And this is sort of a unique layer of the virus that hasn't really been explored, but that they think is very profound and that should be explored further, is what they're saying. So sorry, this one took a very, very long time, but again, this is very dense, very interesting. Sorry that I had to speed read there, but I didn't want to have this end up being like 3-4 hours. So hopefully you all learned something. I learned a bunch of things in this. I thought this was pretty cool. Voila, long COVID is real. Yes, this probably is not like the only part of long COVID, but it's probably a significant part for the immune responsiveness. Like perhaps certain people might have like an overactive immune response to changes to things, or but it's probably just one layer. Long COVID is probably, it obviously is real, but there's probably multiple layers to this because there's also things that could be like viral persistence, which can cause issues and continue generating these things. In addition to um, like a host of other things too as well. Appreciate you as well. But yeah, hope you and everyone else learned something. Yeah, have a good rest of your day. I am going to the gym.